Hello, I'm Fatima Madondo, and today I'll be presenting to you my research in Dr. Peter Didon's lab. My research was on profiling transfer RNA modification patterns in human embryonic kidney cells under oxidative stress. Prior research has shown that cells adapt to stress by reprogramming a system of dozen wobble or third anticodon nucleotide modified ribonucleosides in transfer RNA. This causes selective translation of codon-biased mRNA, representing different classes of critical stress response proteins. My research is to observe if such modifications also occur in eukaryotic cells, specifically in human embryonic kidney 293 cells, which has not been done before when they're exposed to hydrogen peroxide. This will provide insights into the transfer RNA epitranscriptome during cellular stress response and help understand the translational control mechanism. So this slide shows the big picture of my project. Firstly, I exposed human embryonic kidney cells to hydrogen peroxide for 24 hours. Afterwards, I proceeded to isolate and purify transfer RNA using the PureLink kit. The PureLink kit consists of a spin cartridge that allows the separation of transfer RNA into small RNA and large RNA. Large RNA is removed first after centrifugation, and the remaining small RNA is eluted using sterol RNase-free water. Transfer RNA is further hydrolyzed into its nucleoside components to allow further analysis in the LC-mass. The LC-mass profiles the modifications in the transfer RNA sample. The concentration of transfer RNA can be quantified using nanodrop. The nanodrop also shows the purity of RNA nucleosides because RNA absorbs at 260 nanometers. The ratio of absorbance at 260 nanometers and 280 nanometers is used to assess the purity of RNA. A ratio of about 2 is accepted as pure for RNA. I did several experiments on RNA isolation, and some of the results have differences. This could have been caused by pipetting errors in cell lysis volume or by transfer RNA degradation. However, all RNA samples were pure according to the purity ratio shown below. I wanted to check the quality of my transfer RNA samples to make sure there was no degradation. I used the Bioanalyzer for this step. The Bioanalyzer separates transfer RNA by size through a capillary electrophoresis mechanism. Small RNA samples travel faster than large RNA. A marker with standard peaks is used to compare the peaks generated. My results show that my transfer RNA samples had traces of ribosomal RNA and some degradation. Lastly, we used the LC-mass to profile modifications in my samples due to oxidative stress. I hydrolyzed my transfer RNA samples to monoribonucleosides, and they were injected in the LC-mass. The monoribonucleosides have different retention times, and this allows their separation when injected into LC. Some of the compounds take longer to move through the column. There are about 50 non-eukaryotic modifications. The first compartment, or Q1 of the mass spectrometry, selects precursor ions for these non-modifications from the LC-eluted nucleosides. The precursor ions pass through a filter to the second compartment, where they are broken into smaller pieces that are passed on to the third compartment for detection. Fragmentation of the precursor ions allows the separation of ions that have the same molecular weight to be detected, as they are fragmented into different sizes that can be easily differentiated. The results indicated that many wobble modifications, such as MEN-GAL-Q, NCM5U, NCM5U-M and Q, significantly decreased after hydrogen peroxide treatment, supporting that oxidative stress-induced transfer RNA modification reprogramming may take place in human embryonic kidney 293 cells. Further studies are needed to clarify its linkage with stress-essential genes regulation. I would like to thank Dr. Peter Didon for allowing me to work in his lab. I would also like to thank my supervisors Jing San and John Zhou for their unwavering support. I would also like to acknowledge my other lab members who were kind enough to have me around and let me do my work. Thank you.